All right, so hi everyone. So Chris Bradley, I'm CEO and co-founder of Matter Bio. So what I'm going to be talking about today is going to be pretty fast, but it's basically a bit of a combination of this morning where several speakers talked about this concept of entropy and also replacement. And so I'm going to be touching a little bit about that, but from our perspective, which is really sequence integrity of the genome. So this is obviously a very technical audience, but a lot of people think one body, one genome. And we now know, of course, that that's not true. That every cell is different. Every cell in your body is slightly different from the one next to it. It's actually 1 trillion genomes per person, not just one. And this has really important implications. So first of all, how do those changes happen? What does that mean? Well, being alive damages DNA in all sorts of ways, exogenously, endogenously. We know this. It's inevitable. Things hurt the DNA, and sometimes it's not repaired fully. And what happens at that point, amongst many things, at least from a sequence perspective, is the inclusion of noise. And that noise happens in two major ways: point mutations, and then this catch-all structural variants, which is basically larger changes to your genome. And why do we care? Well, lots of data coming out now is showing that there's a tight correlation, not necessarily a causation, but a correlation between these amounts of mutations and lifespan across organisms and within also humans. So they accumulate in a predictable way as we age. And it looks like potentially, once you hit around 5,000, whether you're a mouse, a giraffe, a human, or a naked mole rat, that seems to be the upper bound that you see, at least in the wild, before that animal is dead, at the end of lifespan. And what you see here, this is from Alex Kagan and Inigo Martin-Carena's work. There's a very tight correlation, not just with the total number, which is already interesting, but also the speed at which those mutations accumulate seems to correlate to the speed of the lifespan. So again, a correlation, but a very interesting one. And we know that, of course, that this is also a measure of the damage that occurs over that lifespan. Now, because we're at Foresight, a lot of space talk is coming out recently, and space is a highly damaging place. So if we want to go to Mars, we want to spend any time up in low Earth orbit, we're going to want to know about this because it causes damage and it also accelerates aging. So that said, a lot of these mutations are neutral. So some are good, most don't do anything, and some are bad. So not all mutations are made equal, and they accumulate over time in tissue, and so you start to see this mosaicism. So these are some examples of clonal expansion, tumors, metastases, senescence, apoptosis, so less and less functional tissue. So what do we want to do? The angle we're approaching this from is we want to take that and bring it back to baseline. Bring the genome back to where it started when you were first born. That's fundamentally the engineering problem we want to solve. And we want to do that at the level of genome sequence, so the actual letters. So there's a couple of angles to this. So we have three major strategies. First is reading the genome. Part of the reason this is a relatively new field is seeing these mutations is incredibly difficult. They're random and they're different between each cell. So until recently, normal sequencing does not pick that up, because they don't know if it's an artifact or not. So step one is reading the genome to better understand these mutations and their implications. Removing cells that are what we're calling too far gone, too far corrupted. You don't want to try to rescue cancer cells. Get rid of them. So get rid of tissue that's bad for sure, cells that are toxic. And then finally, renew the tissue that's there. This all seems obvious, but this is really the kind of three-part plan if you're going to want to do this in someone who's already alive. And I would argue potentially in tissue that you want to implant in people as well. So from our talk around replacement, I'd be very curious if this could improve replacement success or efficiency. Okay, so that sounds great. How are we actually doing this ourselves? So as you can imagine, there's some technology behind this. I'll go fast and I'm happy to talk into detail. So we have an approach to amplifying DNA fragments that allows you to actually pick up these mutations in fragments of DNA and differentiate them from mistakes or artifacts. So that's a key step. This is also important for understanding the role, but also knowing if before and after you actually lowered the mutation rate, which you hope in any of these interventions. And so that's one. We have a synthetic bacteria that we've engineered that selectively infects cancer cells. And we are using that as a delivery vector for antigens, but it could be used for all sorts of other things. And this is actually entering phase 1 clinical trials, hopefully at the end of 2024, so in about 6 to 8 months. That might be a little aggressive, but roughly. And then finally, we have several editors, and one of the ones I want to mention today is the ability to now insert very, very large whole genome amounts of DNA into the sequence. And what we think, this obviously has a lot of applications outside of longevity, but for longevity, you could rescue whole genes that you want to make sure are kept at baseline. And then you also want to maybe augment the genome with variants that we know are in centenarians or are somehow protective of DNA damage or of other hallmarks. And that's all I've got. I've got details, but I wanted to keep time short, so I'll stop there.
Thank you. Great. Cool. Let's see if you have any questions. Otherwise, we can go into details.
So very interestingly, we have been publishing a paper that's on the front cover of Aging that's specifically saying biological noise is a way to how to measure aging process. And Irina Comboy was the co-founder of the company. So we use plasma replacement. Right now, we call it as plasma dilution to dilute the senescence, like cytokines and also inflammatory factors in blood. Have you guys considered using this pathway?
Yeah, so like diluting blood of inflammatory factors?
Right.
Yeah, we're hoping that you could prevent those inflammatory factors from being there if you do this well enough, but we're always interested in other strategies. I mean, we're staying away from like protein level or small molecule interventions because we have enough on our plate on just getting this right. But yeah, I think that makes a lot of sense.
Yeah, and which one is the furthest along right now?
The furthest along is this one. So this one is we've spoken to the FDA. We have a full clinical team working on a trial in advanced pancreatic cancer. So yeah, this is ready to go.
Thanks. Thank you. Anyone who hasn't asked a question yet? No? Oh, sorry. Speak up. If I don't see you, just shout it out.
So if you can detect cells that are no bueno, why not just kill the bad ones, and why bother renewing ones that are marginal?
Well, so given a long enough, this is actually really important. So let's assume for a second that this has nothing to do with actual aging, and this is just an accumulation of mutation. It's just a byproduct of what's causing aging, let's say epigenetic aging. They do accumulate, and eventually they are going to do something bad. And the biggest example of that is cancer, right? The multi-hit hypothesis in cancer is generally agreed upon. So like, at some point, you're going to get cancer again. If you live on a long enough timeline, this noise will become overwhelming and will start to create a lot of problems. So,
I mean, the ones you would have renewed, why don't we just kill those too?
well, you're not going to have any left. That's honestly, right? Yeah, so stem cells exhaust themselves. This is actually a good point. So one of the targets, so this is, if you think about it, how are we going to target every cell, right? Delivery is a huge issue. We want to start with stem cell niches because they actually renew the tissues for you. And those are some of the ones that are most implicated in a lot of diseases as we age, like clonal hematopoiesis in hematopoietic stem cells. You run out of stem cells, and the ones that are left over are slowly becoming leukemia and causing all sorts of cardiovascular issues. So there's some evidence that these mutations are disease-causing as we age themselves, and there's no easy way to get around the fact that if your stem cells have them, you're stuck with these mutations. It's a one-way street.
I think you already touched upon it. I was going to ask, how would you target cells if you wanted to replace them? Like, a delivery is what was my question.
Yeah, and we don't have a magic answer to delivery. The only way we're thinking about it is to narrow down the types of cells we're targeting first, do it ex vivo if we have to, but we really want this to be in vivo, and then narrow the area of attack. So we're not going to replace the whole genome or anything close to it. Start with cells that are proliferative or that are rejuvenating the tissue, so stem cells, and then focus on areas of the genome that if they are corrupted, if they are mutated, cause the most harm, and then go backwards from there. And then, yeah, if someone has a systemic delivery that's highly efficient, I'll use that.
Which stem cell niche? Because most stem cell niches are very difficult to get them back and then engraft. So
if I were to pick one, the absolute lowest hanging fruit, which is still difficult, would be HSCs. Because there's also indications you can use for the FDA. So like clonal hematopoiesis and myelodysplastic syndrome specifically.
Thank you so much.
Yeah, for sure.
All right, awesome. Thank you so much.